Hello everyone. Today I'd like to tell you about spatial gene expression patterns in the intestine, and this is a project that I worked on with my partner, Rita Manko, at Shalev lab at the Weizmann Institute. So the small intestine is where most of our food absorption happens, and it's also an interface with the outside world, making this tissue even more interesting. So the intestinal epithelium has the shape of these protruding finger-like villi, which increase the surface area of the intestine, and thus this increases absorption. If we zoom into one single villus, we see that at the bottom, we have these stem cell containing crypts, and they're responsible for renewing the intestinal epithelium. The stem cells inside the crypt divide, and as they divide, they push the differentiated cells upwards, and they travel along this sort of conveyor belt up towards the tips, and then in a few days, they're shed off, and that is how the tissue is renewed. So 90% of the cells which comprises epithelium are absorptive enterocytes, which absorb nutrients, but they're not alone. Other rare cells also perform crucial functions for the intestine to operate. So these cells contain goblet cells, which secrete protective mucus, Tuft cells, which are chemosensory, and enteroendocrine cells, which secrete hormones into the lumen. As a cell travels from the crypt to the tips, they're exposed to an ever-changing environment. They're exposed to gradients of nutrients, oxygen, microbiome exposure, and more. But what about the cells themselves? So previous work from the Itzkovich lab has shown that enterocytes, the absorptive cells, they drastically change their transcriptome as they travel from the crypt to the tip. And the way they did it was by performing laser capture microdissection from 5 distinct areas along this trajectory. And sequencing these samples, they arrived at landmark genes, which mark the location for the enterocytes, and then were then able to take single-cell RNA-seq data and map them back to the tissue using the landmarks, and thus reconstructing the whole spatial transcriptome for enterocytes from the crypt to the tip. And they were able to see that the vast majority of genes expressed by enterocytes are indeed zonated, as we like to call it. And this got us very interested in what happens in the rare cells. Do they also express these spatial zonation patterns? And how is the spatial division of labor work in the rare cell types? However, we could not use the same method since in laser capture microdissection, the very rare secretory cell-derived transcript would have been simply diluted out. We had to come up with something else to see the zonation patterns in our rare cells, and that is why we developed ClumpSeq. So ClumpSeq starts with non-ideal dissociation of the tissue, and this gives rise to clumps of cells instead of single cells, and these clumps are 2 to 10 cells large, and due to the fact that they all contain enterocytes, we can know which part of the tissue they came from using the landmarks from the previous paper. So if we then sequence these clumps of cells, we also have inside them the rare secretory cells, and we can use the known location from the enterocytes to infer secretory-specific landmarks. And then we can take data sets of single cells, of secretory cells, and again map them back to our tissue and reconstruct the entire transcriptome, the spatial transcriptome, based on single cells for our rare secretory cells. And when we applied this method to goblet cells, you can see here, we reconstructed the whole transcriptome, and we saw that a significant portion of goblet cell genes are indeed zonated. We then validated our reconstruction using single molecule FISH for some genes, you can see here, and we could then look at the functions. So goblet cells, their main function is secreting mucus. And when we looked at this heat map of the spatial patterns of the genes that participate in the composition of mucus, we saw that mucus actually changes gradually from the crypt all the way towards to the tip. We then performed gene set enrichment analysis to see what other interesting things are happening for goblet cells, and we saw that there is an enrichment of immune-related programs at the tip for goblet cells, such as interferon gamma signaling, inflammatory, and so on. And we then validated this finding by looking at IDO1, and we indeed saw that it is strongly zonated towards the tip of the villi. Now shifting our focus onto Tuft cells, I'd like to start by a brief introduction. So this paper from Haber-Bitton et al. From Aviv Regev's lab at the Broad has discovered that Tuft cells actually differentiate into two distinct subpopulations, and they named them Tuft1 and Tuft2. One population performs functions that are related to the neuronal nervous system, and the other population performs immune-related functions. And when we looked at the marker genes for these populations in our spatial reconstruction, we saw that not only are they functionally separated, they're also spatially separated, with the neuron-related Tuft1 cells concentrated towards the crypt and the immune-related Tuft2 cells towards the tip. So again, we see an enrichment in immune-related function towards the tip, not only in Goblet, but also in Tuft cells. We also saw some interesting metabolic-related functions. For instance, this is fatty acid binding protein 1, which is zonated towards the villus tips. And last but not least, enteroendocrine cells. So in this seminal paper from Hans Clever's lab, they have reconstructed the temporal expression patterns for enteroendocrine cells in the intestine. And when we took their single cell data and we ran it through our computational pipeline, we now had both space and time. And these should correlate because as cells age, they also exit the crypt and travel towards the tip. And you can see in these several examples that indeed there is a good agreement between the spatial and the temporal profiles. And when we did it systematically for all the genes, and each point here in this plot is a gene, and you can see on the x-axis its central point in spatial expression and the center in temporal expression, and most parts these correlate. But we notice here in the upper left part of the plot that there is this group of genes for which the temporal and spatial patterns do not correlate. And they are in the crypt, expressed mainly in the crypt, but they are expressed late in a cell's life. And you can see here the discrepancy between the spatial and temporal patterns up here for these genes. And I would like to point out that for enteroendocrine cells are actually comprised of different subpopulations, and each subpopulation specializes in its own cocktail of secreting hormones. And the way to settle this difference, this discrepancy between the spatial and temporal patterns would be to notice that the cells that express these specific hormones, they actually migrate out of the crypt much slower than the others. So during late times in their lives, they express these genes, but they're still very close to the crypt. And we validated this hypothesis for somatostatin expressed by delta cells. And you can see here in these images and this quantification of the single molecule fish that somatostatin expressing cells are actually enriched in the crypt and villus bottom compared to the middle and the tip of the villi. So now to summarize, I've shown you ClumpSeq, which is a general approach to reconstruct zonation of rare tissue cells. It can be also applied to lungs that contain goblet and tuft cell as well, to spatial zonation in beta cells in the pancreas, to kidney nephrons and other tissues. Also, I've shown you that rare secretory cells in the small intestine exhibit functional spatial division of labor with specifically immune-related functions, which are enriched in the tips of the villi, and that in enteroendocrine lineages, there are heterogeneous patterns of migration out of the crypt and towards the tip. And finally, I'd like to thank my partner in crime, Rita, our wonderful mentor, Shalev, all the others that worked with us, the funding sources, and the organizers for giving me this opportunity, and all of you for listening to the talk.
Thank you very much, Ina. Great on time, so I think we'll have a little more time for questions. Do we have any questions from our panelists? And if not, I can read from our Q&A. So Theodore K. Plus is asking, yes. Oh, sorry.
Oh,
yes.
I have a question.
Hi.
Hi. So, Ina, the fact that you're with Shalev, does that mean that you're not looking for a job, or you are looking for a job?
Currently, I've started a second postdoc in Stanford. I did a brief postdoc in Shalev's lab as a bridging postdoc before I started the current one in Michelangelo's lab in Stanford. And also, before you ask, this work is under review.
Okay, so the scientific question, why do you have the immune genes or patterns in the tip? What is this obvious?
I don't know if it's obvious, and we're not sure why that is, but this could have to do with larger exposure at the tip to microbiome or possibly to external organisms.
Mhm. Okay, thank you.
Thank you.
Great, thank you. So we have a question from the audience, from Theodore. What is the advantage of ClumpSeq over published methods, for example, SlideSeq, or even commercially available spatial technologies such as 10x genomics, which can identify the location of single cells on a tissue?
So the first advantage is resolution. This is single cell based because we do map single cells to their precise location in the tissue. When compared to SlideSeq, for instance, every method that is not single cell based but uses these small bulks like laser capture, microdissection, or SlideSeq might suffer from the same issue, which is the dilution of transcript that come from rare cell populations. And the only way to prevent that is having higher resolution, such as actually single cell based method of reconstruction.